Down here, glowing can make you harder to see. That sentence should be wrong. Light attracts attention. Anyone who has held a torch in a dark room understands this instinctively. But roughly 600 meters below the ocean surface, a small silver fish called Argyropelecus affinis has turned that assumption inside out, and the marine biologist E.J. Denton first measured exactly how in 1972. The hatchetfish's problem is geometric. Any predator hunting from below sees its prey as a dark silhouette against the faint, diffuse glow of sunlight filtering down from above. The fish is a shadow, visible, edible. Argyropelecus solved this by evolving rows of photophores, light-emitting organs, running along its belly. Those photophores emit downward at precisely the same brightness and color as the downwelling light above them. Blue at around 480 nanometers, which happens to be the wavelength oceanic water transmits most efficiently. When a predator looks up, the fish's silhouette fades into the background. The animal glows and, in doing so, disappears. Denton's 1972 paper in the Proceedings of the Royal Society B measured the angular distribution of emitted light from Argyropelecus affinis ventral photophores across two vertical planes. His conclusion was that the distribution was of the kind required to match, over a wide range of angles of viewing, the external daylight field in the ocean, and that these observations gave strong support to the hypothesis that the function of many photophores is to give camouflage. A 2017 paper by Feller and colleagues in the same journal added a further layer, a novel two-component photonic structure in hatchetfish skin itself, superficial stacks and deeper elliptical bundles that scatter incoming sideways light downward through the belly photophores. The skin routes lateral illumination into the counter-illumination system. Three-dimensional camouflage, not just ventral. This requires active management. The fish must track the changing ambient light as it moves through the water column and adjust its output accordingly. Young's 1977 shipboard aquarium experiments demonstrated this directly. Midwater animals placed under adjustable overhead illumination turned their photophores on in response to light, off when the light was extinguished, and regulated intensity across a wide range in between. Cronstrom and colleagues, in a 2005 Journal of Experimental Biology paper on Argyropelecus hemigymnus, identified nitric oxide as the specific neurochemical pathway controlling photophore output in real time. A 2020 Proceedings of the Royal Society B paper by Davis and colleagues added one more layer. Many deep-sea fish possess small photophores oriented inward, facing the eye rather than outward. These appear to function as light meters. The fish samples ambient downwelling light through this internal reference and calibrates its belly glow accordingly. It's a feedback loop built into the anatomy. One honest caveat: no study has placed a live predator beside a counter-illuminating hatchetfish and directly measured detection rates. The camouflage function rests on optics, physiology, and modeling, not on live predator-prey behavioral experiments. The optics are convincing. The inference is well supported. But the distinction is worth holding. In Subnautica, the deep ocean is threaded with glowing organisms, creatures that pulse and shift and flicker in the dark. Players tend to register that glow as atmosphere. The biology says it's never atmosphere. It's always doing a job. The hatchetfish's light conceals one animal. The next function conceals something else entirely, the predator behind the light. At the end of a modified dorsal fin spine, the elysium, extending from the head of a female Melanocetus johnsonii, a small, faint blue-green light hangs in absolute darkness. The humpback anglerfish, family Melanocetidae, inhabits depths from 100 to roughly 1,500 meters across tropical and temperate oceans worldwide. She has a jaw that can hinge wide enough to swallow prey approaching her own body mass, enlarged backward curving fangs to prevent escape, and at the tip of that spine, a structure called an esker, a bag of glowing bacteria. That last detail took decades to confirm properly. The light in an anglerfish's esker isn't produced by the fish itself. It comes from bacterial symbionts living inside the lure. A colony Haygood, distal and herring quantified in 1992 had approximately 400,000 cells packed into modified tissue using 16S rRNA analysis of juvenile M. johnsonii specimens. Older, popular accounts attributed this light to bacteria from the genus Photobacterium, but a 2019 eLife paper by Baker and colleagues sampling ceratioid anglerfishes from across the Gulf of Mexico identified the M. johnsonii symbiont as a distinct provisional species, Candidatus enterovibrio escacola. These bacteria have never been cultured outside the esker. Their genome shows progressive reduction consistent with long-term specialization inside a single host tissue. The fish acquires them from the surrounding water, not from its parents. 9 of the 11 ceratioid anglerfish families carry a bioluminescent lure. All 9 appear to use this same environmental acquisition route. The bacterial colony glows continuously. The fish controls how much light escapes by covering and uncovering the esker with muscular pigmented tissue and by regulating oxygen supply to the bacteria. The result is an adjustable signal, steady, flickering, pulsing. A reflective cup of flat cells and guanine crystals amplifies and directs the emission. Whether prey actually approach and are captured at the lure hasn't technically been confirmed by direct observation. No footage documents a prey animal investigating an anglerfish esker and being struck. The hunting function is inferred from anatomy. The lure positioned directly above a gape wide enough to engulf whatever investigates. Stomach contents showing diverse midwater prey. The straightforward logic of a moving light source in a lightless environment where anything luminous is worth investigating. The esker also appears to serve mate detection. Dwarf males locating females across 3-dimensional darkness guided by the same signal. 1 structure, 2 functions. No confirmed footage of either. The inference is strong. The gap is real. Subnautica players who spent time watching glowing lure predators, creatures that dangle an attractive light ahead of a much larger hidden mouth, were watching a functional reconstruction of this feeding strategy. Bioluminescent lures evolved once within a derived deep-sea ceratioid lineage during the Oligocene. 1 evolutionary event, then 11 family radiations. The lure is ancient. The light that looks like an invitation is a death trap that evolution invented once and kept forever. The hatchetfish glow hides the animal behind it. The anglerfish glow hides what kind of animal lies behind it. Both use deception through light. The third function abandons deception entirely and operates more like surveillance. 3 genera of deep-sea dragonfishes, Malacosteus, Aristostomias, and Pachystomias, all within the family Stomiidae, produce something no other vertebrate produces: far-red bioluminescence. Not the blue-green emission that virtually every other bioluminescent deep-sea creature generates. Not ambient dim amber. Proper red light, at wavelengths peaking around 705 to 710 nanometres from their suborbital photophores, first measured and confirmed in a 1984 paper in Science by Edith Widder, Michael Latz, Peter Herring, and James Case. The paper was titled Far-Red Bioluminescence from 2 Deep-Sea Fishes, and it placed the suborbital photophore peak of both Aristostomia scintillans and Malacosteus niger at approximately 705 nanometers using spectrophotometry. Before that paper, the far-red end of the spectrum in deep-sea fish had been theorized but not measured directly. Widder went on to become one of the most respected bioluminescence researchers in the world, co-founding the Ocean Research and Conservation Association, and the 1984 measurement remains the foundation of this entire line of inquiry. One physical fact makes the significance of far-red light in the ocean immediately clear. Seawater absorbs red wavelengths rapidly. Below roughly 30 meters, The red end of the visible spectrum has been stripped from sunlight almost completely. The deep ocean is a world of blue and black. Any animal emitting in the far red is broadcasting a wavelength that effectively does not exist in the ambient environment, and that most of the animals in that water can't detect. The vast majority of deep-sea fish have visual systems maximally sensitive in the narrow blue-green window between 470 and 490 nanometers. These animals have no receptors tuned to anything above roughly 600 nanometers. For them, the dragonfish's suborbital photophore does not exist. The fish illuminates the water in front of it with a waveband invisible to most of its prey. A 2023 mitochondrial genome paper on Malacosteus niger, published in Zookeys, described the species' suborbital photophore in plain terms. It functions as a private torchlight to detect prey items unable to see red light. The fish lives between 600 and 1,000 meters depth. It hunts under illumination only it can use. The mechanism behind the red emission is itself worth a moment. Malacosteus niger does not produce red light directly. The initial bioluminescent reaction generates blue-green photons in the standard way, luciferin oxidized by luciferase. Those photons are absorbed by fluorescent proteins within the photophore, which re-emit at longer wavelengths. A brown outer layer of the photophore filters the output further, shifting the final emission into the far red, around 705 to 710 nanometers. Remove that brown layer in a laboratory preparation, and the emission shifts back to approximately 650 nanometers. The entire structure is a biological frequency converter. The fish takes blue-green chemistry and ends up with near-infrared output. The visual system that lets M. niger see its own emission is equally unusual, and this is where the three genera diverge. Aristostomias and Pachystomias evolved actual long-wave shifted visual pigments, opsins absorbing at approximately 515, 550, and 590 nanometers. Their long-wave sensitivity is extended through multiple shifted pigments working in combination. Malacosteus niger took a different route. Its retina contains only two conventional rod pigments, peaking at approximately 520 and 540 nanometers, comfortably short of the far red. A 1999 vision research paper by R.H. Douglas and colleagues showed that the fish compensates through a photosensitizer, a derivative of bacteriochlorophyll C and D, distributed throughout the retina, which absorbs strongly at around 673 nanometers and transfers that energy to the visual pigments. A 2016 Scientific Reports paper by Douglas, Jenner, Hudson, Partridge, and Wagner confirmed the photosensitizer is present across the entire retina, with absorption peaking near 673 nanometers and fluorescence emission around 680 nanometers. An astaxanthin-based retinal tapetum reflects long wavelength light back through the photoreceptor layer for a second pass, further boosting sensitivity. Where does the bacteriochlorophyll derivative come from? The fish eats it.
Stomach content analysis has shown that Malacosteus niger, despite its enormous fangs and a gape almost a quarter of its body length, feeds on mesopelagic copepods. Those copepods contain bacteriochlorophyll-derived pigments, whose fluorescent spectra match the retinal photosensitizer almost exactly. M. niger builds its private visual channel by eating the correct prey. Red vision in the dragonfish lineage evolved approximately 15.4 million years ago, according to Kennelly and colleagues' 2013 comparative innovation study, and it evolved once, with the three genera then diverging in the mechanisms they use to maintain it. The range of the red searchlight is short. Far red wavelengths are absorbed by seawater within meters. This isn't a long-distance beacon. It illuminates what is immediately in front of the fish, invisible to the prey until the teeth arrive. Two calibrations belong here. First, a small number of lanternfish species, among them Myctophum nitidulum and Bolonycthis longipes, carry long-wave shifted sensitivities sufficient to detect stomide far-red bioluminescence from up to approximately 7 meters away. The private channel has eavesdroppers. Second, the searchlight function, while supported by multiple peer-reviewed spectral and visual physiology papers, rests on inference. Direct observation of Malacosteus niger hunting by red light in its natural habitat does not exist. The photophysics are solid. The behavior follows from them, but the record is careful to note the distinction. The deep sea had, incidentally, solved RGB illumination approximately 15 million years before anyone built a gaming peripheral. The dragonfish was here first. 3 functions. A glow that erases the glowing animal. A glow that conceals a predator behind an invitation. A glow that illuminates prey in a waveband only the predator can see. The 4th function reverses all of that directionality. It deploys bioluminescence against another organism, using light to expose rather than hide. The formal name is the burglar alarm hypothesis, and it was first proposed by L. Birkenrode in 1943 to explain why marine dinoflagellates, single-celled organisms roughly the size of a grain of sand, produce brief flashes of light when physically disturbed. The logic of flashing seems backwards. Dinoflagellates can't escape. They don't have venom, or speed, or armor. Flashing under attack should only make them easier to find. Birkenrode's proposal inverted that reasoning. The flash isn't aimed at the thing eating the dinoflagellate. It's aimed at whatever is above the thing eating the dinoflagellate. The flash illuminates the grazer, copepod, amphipod, and makes it visible to visual predators hunting from higher in the water column. The dinoflagellate can't flee, but it can call for help. In 1993, Mark Abrahams and L.D. Townsend published an experimental test of this hypothesis in the journal Ecology, titled Bioluminescence in Dinoflagellates, a test of the burglar alarm hypothesis. They placed luminous and non-luminous dinoflagellates, grazing copepods and 3-spined sticklebacks together in glass jars and measured predation rates. Copepods were eaten at significantly higher rates in the presence of luminous dinoflagellates. The flash functioned as Birkenrode predicted. The dinoflagellate's distress signal attracted secondary predators onto the primary one. The same year, Fleischer and Case demonstrated that squid could use dinoflagellate bioluminescence to locate and capture non-luminous prey in the vicinity, confirming the light's utility as a hunting aid for a third party. Edith Widder has described the underlying dynamic in plain terms. The best chance you have when you're getting attacked is to attract something bigger than what is eating you. Vampiroteuthis infernalis. The same logic extends upward into larger animals.
Vampiroteuthis infernalis. The vampire squid, which is neither a vampire nor a true squid, but occupies its own order, Vampiromorpha, formerly described by Carl Chun in 1903, has no ink sac. When threatened, it ejects a sticky, bioluminescent mucus from the tips of its arms. The behavior was confirmed by direct observation of live specimens captured by ROV and studied in laboratory settings, documented in the Biological Bulletin in 2003. The luminescent cloud does not mask the squid's silhouette the way ink would. It illuminates the attacker, potentially making the predator visible to its own predators. The vampire squid does not escape predation by outrunning it. It reverses the exposure. Subnautica's bioluminescence reads like aesthetics. Players encounter glowing plankton that shift color near different light sources, predators flanked with biolamps, environments that seem to pulse with ambient light as a design choice. The game's own wiki, writing about bioluminescence in the Subnautica universe, acknowledges it drew on real functions: counter-illumination, prey luring, predator confusion. Whether any specific game creature was confirmed by its developers as modeled on a named real species isn't something the documentary record establishes. No developer statements on record make that specific claim. But that ambiguity is itself revealing. The game didn't need to be a precise biological reference to capture the functional logic, and players felt it anyway. The glow in Subnautica behaves the way deep ocean glow actually behaves. It's never decoration. It's always working. The deep sea didn't discover one way to use light. It discovered dozens,
and then, across hundreds of millions of years, refined each of them into a precision tool tuned to a specific problem in a specific physical environment. Counter-illumination solves the silhouette problem. The lure solves the problem of prey that won't approach. The red searchlight solves the problem of prey that can see you coming. The burglar alarm solves the problem of a predator you can't outrun. Argyropelecus affinis glows and disappears. Melanocetus johnsonii dangles a light and waits. Malacosteus niger sweeps a near-invisible red beam through the darkness in front of it, searching for copepods it will illuminate before they know they are being hunted. And the dinoflagellate flashes once, briefly, and hopes something larger noticed what was eating it. The same basic chemistry, luciferin oxidized by luciferase, or a variation close to it, producing camouflage, predation, illumination, and an alarm signal, all within the same 600-meter column of water. One phenomenon, 4 functions, all of them contradicting the assumption that a glow-in-the-dark is simply a glow-in-the-dark. Subnautica felt alien because the real ocean already was. Which real use of deep-sea light feels most like Subnautica to you? The camouflage, the lure, or the private searchlight? Drop it in the comments. If you want more stories like this, subscribe